Hello, grade 10. So we are still in section 4, genetic engineering of chapter 9. We said yesterday that DNA sequences of organisms can be changed through a technique called genetic engineering. First of all, let's remember what is cloning. We say a clone is a genetically identical copy of a gene or of an organism. And cloning occurs in nature through asexual reproduction in different types of organisms like bacteria, some plants, and some simple animals like the hydra and the yeast by budding. Mammals cannot be cloned naturally. They can be cloned through a process called nuclear transfer, in which a nucleus is removed from an egg cell of an organism, and a nucleus of a cell from an animal to be cloned is implanted in the cell. As you see here, this is an egg cell of one, let's say one sheep, okay, and this is a cell, body cell of the sheep that want to be cloned. They take the nucleus out from this body cell with its DNA, and they insert it in the egg cell. They remove the nucleus of the egg cell and they insert this nucleus in the egg cell. Then the egg cell is stimulated. After it starts dividing and the process is successfully happened, they will be implanted in another female to produce the cloned organism. Now, cloning has potential benefits, advantages, and disadvantages at the same time. The advantages of cloning could be organs for transplant into humans. They use organs from cloned animals, and these organs can be transplanted into humans. Also, cloning can be used for saving endangered species of different organisms. The disadvantages of cloning are low success rate. In addition, clones are less healthy than the original animal because of premature aging and old DNA, as well as the decreased biodiversity, because if they clone endangered species, this will decrease the biodiversity among the living things. And there is a tension here you have to keep in mind. A clone may not look like or behave like the original because, as you learned, many factors include the environment affect the expression of genes. Now, the genetic research relies on cloning for genetic engineering, but not cloning the organism. Instead, it is cloning of individual genes. A clone of a gene is a copy of that one segment of DNA. In some cases, scientists insert cloned genes from one organism into a different organism. So, new genes can be added to an organism's DNA. And genetic engineering in this way involves changing an organism's DNA to give it a new trait or a desired trait. Genetic engineering is based on the use of recombinant DNA. And recombinant DNA means that it's a DNA that contains genes from more than one organism. As you can see here, this is a plasmid DNA loop from the bacteria. Here, there is a gene that is inserted with the plasmid of the bacteria. This gene has a DNA sequence from another organism. So this becomes a recombinant DNA. And that means DNA that has genes from more than one organism. This one from the bacteria, the brown one, and the blue one from another organism. Now, bacterial plasmids are often used to make recombinant DNA. So they depend on bacteria in genetic engineering because they have plasmids, and these plasmids are enclosed loops of DNA that are separate from the bacterial chromosome, and they have the ability to replicate on their own within the cell. Now, what they make is they use the restriction enzyme. This restriction enzyme, they have the same restriction enzyme that cut the plasmid DNA and the foreign DNA that want to be inserted, okay? Then, they stick together the gene from the foreign DNA to the plasmid DNA, and at the end you will have the recombinant DNA here, which contains of genes from different organisms. Now this plasmid will be inserted into the bacteria again, and this bacteria will be transformed bacteria with new genes. Because of this, we call it transgenic organism. It becomes a transgenic organism. It has one or more gene from another organism inserted into its genome. So referring to